Hello, welcome to Biology, the scientific study of life. RNA is viral genetic material. Although more phages and mastoplans viruses are RNA viruses, the broadest variety of RNA genomes is found among the viruses that infect animals. Among the three types of single-stranded RNA genomes found in animal viruses, the genome of a class, this class 5 class, notice that viruses can directly serve as mRNA, and those can be translated into viral protein immediately after function. This figure shows a virus of a class V in which the RNA genome serves as a template for mRNA synthesis. The RNA genome is transcribed into complementary RNA strands, which function both as mRNA and as template for the synthesis of additional copies of genome RNA. Like all viruses that require RNA to RNA synthesis to make mRNA, this one uses a viral enzyme that is packaged with the genome inside the capsid. The RNA animal viruses with the most complicated reproductive cycles are the retroviruses, class 5 class, notice that this one. These viruses are equipped with an enzyme called reverse transcriptase, which transcribes an enzyme template into DNA, providing an RNA to DNA information flow. The opposite of the usual direction, this unusual phenomenon gives rise to the name retroviruses. This unusual or retro means backward of particular medical importance is HIV, human immunodeficiency virus. The retrovirus that causes AIDS, acquired immunodeficiency syndrome, HIV, and other retrovirus are enveloped viruses that contain into identical molecules of single-stranded RNA and two molecules of reverse transcripts. After HIV enters a host cell, its reverse transcripts molecules are released into the cytoplasm and catalyze synthesis of viral DNA. The newly made viral DNA then enters the cell's nucleus and integrates into the DNA, a chromosome. The integrated viral DNA, called a provirus, never leaves the host's genome, remaining a permanent resident of the cell. Unlike a prophage, a provirus never leaves. The host's RNA polymers transcribes the proviral DNA into RNA molecules, which can function both as mRNA for the synthesis of viral proteins and as genomes for new virus particles released from the cell. Notice this figure. HIV entering a cell. Notice that this one, HIV, and this represents membrane of white blood cell. Notice picture 2, HIV entering a cell. Notice 3 here. Notice that, and HIV, a new HIV leaving cell here, the last one. Closer look, notice that the reproductive cycle of HIV, retrovirus. The photo on the left here, artificial colored TMS, reveal HIV entering and leaving a human, white blood cell. Note in step 4, this step, that DNA synthesized from the viral RNA genome is integrated into the host cell chromosomal DNA, a characteristic unique to retrovirus. Notice that. Notice the steps also. Step 1, the virus fuses with the cell's plasma membrane. The capsid proteins are removed, releasing the viral proteins and RNA. Steps 2 also reverse transcripts catalyzes the synthesis of a DNA strand complementary of the viral RNA. Step 3 reverse transcripts catalyzes the synthesis of a second DNA strand complementary to the first. Another step, the double-stranded DNA is incorporated as a provirus into the cell's DNA. Notice this step also proviral genes are transcribed into RNA molecules which serve as genome for the next viral generation and as mRNAs for translation into viral proteins. Notice these are the viral proteins here. The viral proteins include capsid proteins and reverse transcripts made in the cytosol and envelope a glycoprotein made in the ER. Notice here also vesicles transport glycoproteins from the ER to the cell's plasma membrane. Notice. This step also capsids are assembled around viral genomes and reverse transcript molecules. Notice the last step here. New viruses bud off from the host cell. Evolution of viruses. Viruses do not really fit our definition of living organisms and isolated viruses in biological inert, unable to replicate its genes to regenerate its own supply of ATP, yet it has a genetic program written in the universal language of life. Do we think of viruses as nature's most complex molecules or as the simplest forms of life? Either way, we must bend our usual definitions, although viruses are obligate intracellular parasites that cannot reproduce independently. Their use of the genetic code makes it hard to deny their evolutionary connection of the living world. How did viruses originate? Because they depend on cells for their own propagation, viruses most likely are not the descendants of precellular forms of life, but evolved after the first cells appeared, possibly multiple times. Most molecular biologists favor the hypothesis that viruses originated from fragments of cellular nucleic acids that could move from one cell to another. Consistent with this idea is the observation that a viral genome usually has more in common with the genome of its host than with the genome of viruses infecting other hosts. Indeed, some viral genes are essentially identical to genes of the hosts when on the other hand, recent sequencing of many viral genomes has found that the genetic sequences of some viruses are quite similar to those of seemingly distantly related viruses, such as an animal viruses and a plant viruses that share similar sequences. This genetic similarity may reflect the persistence of groups of viral genes that were evolutionarily successful during the early evolution of viruses and the eukaryotic cells serving as their hosts. The origin of viruses is still a topic of much debate. Perhaps the earliest viruses were naked bits of nucleic acids that made it from one cell to another via injured cell surfaces. The evolution of genes coding for capsid proteins may have facilitated the infection of undamaged cells. Candidates for the original sources of viral genomes include plasmids and transposons, genetic elements. Plasmids are small circular DNA molecules found in bacteria and also in yeasts, which are unicellular eukaryotes. Plasmids exist apart from the cell's genome, can replicate independently for the genome, and are occasionally transferred between cells. Transposons are DNA segments that can move from one location to another within the cell's genome. Those plasmids, transposons, and viruses all share in an important feature. They are mobile genetic elements. The ongoing evolutionary relationship between viruses and the genomes of their host cells is an association that makes viruses very useful model systems in molecular biology. Knowledge about viruses also has many practical applications, since viruses have a tremendous impact of all living organisms through their ability to cause a disease. Thanks for listening.